Welcome back, everyone, to part 2 of our talks this afternoon. It's with great pleasure that I welcome Professor Nick Wheelhouse to the stage. Nick is a Professor of Comparative Infectious Disease and contributes to the leadership of the Centre for Biomedicine and Global Health here at the university. Nick is going to talk through his work, which I'm sure will cover the diagnosis of immunopathogenesis and epidemiological work around about reproductive sciences. But what I want to highlight is Nick has joined us from the Morden Research Institute and is now part of the Napier family, well embedded. Nick is well known beyond the university and currently is the serving interim chair of the Food Standards Agency's Committee on Animal Feedstuff. Nick is also a trustee of the British Society for Animal Science and a fellow of the Higher Education Academy. So we're really pleased, Nick, to hear about your journey to professorship, and the stage is yours.
Thank you. I've got to say this is a first. This is the first time I've ever been in a room with Gary where I am wearing the waistcoat. So it's a day of firsts. So thank you very much. So I did struggle quite considerably with this title. I think many people might say I'm a jack of all trades, and many people will probably agree that I am master of none. But my work really has encompassed, I hope, and it comes across today, nearly every level, right the way from microbes all the way through to some of the work that I'm doing now on planetary health. Just a little bit about myself. So I live down the road, but I'm not originally from down the road. I am a proud Yorkshireman, okay? And for that, I have probably a number of attributes that are consistent with those guys. I'm sheer bloody minded, and maybe some people would use the term pig ignorant occasionally. I think I would also say probably loyal, especially to people sniggering in the front, to a fault. Now I come from north of Leeds, and I highlight this couple of little points here because many people might not know I was brought up in pubs. So just to link back to the previous one, I have a long experience, probably from the age of 0, of my liver being challenged. And hopefully it will make its way back to health at some point in my life. And for the first 15 years, it was in the Fox. But I really highlight these because some strange twist of fate. For the last 2 years of my life before I went to university, we were in the Brown Cow in Bingley, which was our colleague's favourite pub, Dave Minch's favourite pub during his PhD. We didn't mix there. There was about 10 years difference, but it is one of those strange twists of fate that I thought might highlight. As any proud Yorkshireman knows, there is only one main religion, and that is cricket. This is a contemporary picture. It's not when I was smaller. This is my proud day when Harry made his debut for Marchman at Cavalry Park. But it's not the only sport that I played. And I did try and look for embarrassing pictures of myself when I was younger. And I did manage to find one. It doesn't work, but if anybody can look in the front row there, first from my right, there you will see somebody who looks quite smug with an extreme amount of product in their hair, tellingly from the early 1990s. And I can tell you I could play 80 minutes and that hair would not move. Now, sport taught me a lot. It's not just a random picture that I thought I'd put up there. It taught me many things. How to win. Most importantly, as a scientist, how to lose, how to fail, and how to get up again and get up for the next match. And I think it's really important. The other thing was that I've always been a bit of an all-rounder. All-rounder at cricket. I played multiple positions at rugby, prop, second row, and I have to be careful, flanker as well. So, so it's something that I took on in my career and my education. I left school and I did an applied biochemistry degree, and that was something I really felt I needed to do. I needed to do something applied. I couldn't do a pure science. I needed to know what I was doing and why I was doing it. And it's been really important for me that all through my career, I've not really been a fundamental scientist. I've really wanted to be applied in how I see problems. I learned two things. I learned a lot of things during my undergraduate degree. I hate to say two things, but two main loves came out of that degree. One was a love of this place. It looks nothing like this anymore. It's St James's Park. This was St James's Park 1992, when I used to stand on the Gallagher J watching Special K's promotion season there. The other one was a love of laboratory science. And I hate to say it, that wasn't because of my degree program. That was because my tutor invited me one summer to do a job for him. He asked me to do a job and analyse a feed trial, the data of a feed trial in Jersey cows. So it's not just a random picture of a Jersey cow. And it was that experience, being left in the laboratory, not being time managed like we do in lab classes, being able to mix with the postdocs and the PhD students and just be able to ask the questions I wanted to ask, that made me fall in love with science. And because of that, before I'd finished my degree, I was already accepted to do a PhD at the University of Reading. That didn't actually last very long. It's not that I quit, it's because my boss quit. And my boss got a job as head of agriculture in Aberdeen. So as any good PhD student does, you travel with your supervisor. So January, believe it, dragging me up to Aberdeen in January 1996, I started working in Aberdeen with the University of Aberdeen. And back then the route was separate. So it was a PhD between the two. And that was to work, and you'll see lots of different species pop up here, on sheep. So I know I'm in the Centre for Biomedicine, but you'll see quite a lot of ruminant animals appearing throughout this. And eventually we settled on that great title. I mean, it's, you know, it's catchy, eh? But essentially, the key thing for that was that liver cells, we're very proud of this because we managed to be the first people to figure out a way of culturing liver cells from abattoir material. So 7 o'clock every Tuesday morning at Macintosh Donald down the road in Aberdeen, we'd be down there and somebody would slap a liver into my hand. And I would do lots of prep work and I would isolate the cells. And my supervisor was incredibly encouraging throughout my PhD. Anybody who knows him will recognize this kind of comment. And this was the kind of comment when we finally got things working. What do you mean something worked? And it was when we'd actually did a key experiment. And my experiments were basically, I added hormones. Growth hormone's a hormone it causes growth. It does exactly what it says on the tin, but it's made by the brain, but it works through an intermediary that's produced by the liver. And we showed that actually if you change the culture media, if you change the conditions the cells were in, it changed how they responded to that. And we did some subsequent work on that, and we showed that specific amino acids were actually targeting, signaling for this. And it was really quite an important piece of work. I was very proud of this work. And that led to a postdoc at the old medical school in Edinburgh. I was in the 1960s extension around the block, but it's not quite as attractive as that picture. And that was working on, strangely, liver. Because it turns out there's not that many people in the world back then who could actually isolate liver cells alive and culture them, even though I was moving into mice. And Donald Trump's favorite transgenic mice we were working with back then. But this time we were working on something different. We were working on liver cancer, and we were working on the fundamental causes of liver cancer, and we were working on inflammation and liver cancer, actually following on from Lenny's talk there. And this time we were looking at how these molecules made by the immune system cause inflammation, this inflammatory response. actually causes DNA damage. So it directly damages our cells. And I will put this paper up here. I did want to put the data from the paper, but it's behind a paywall. It's that old. You can't actually, we don't have preprints online. And we did this work, and it was really important. It was really important not just for this, but actually it led directly to my next job and place of work, which I was for 11 years at the Morden Research Institute, working with these guys. Gary, that a lot of people will know, and David. And I say sheepish because I wasn't specifically working on sheep. I was working on a bug that infects sheep. And there was a reason why I had a particular skill set that they wanted. One, I'd worked with sheep before. That's helpful. There's a lot of challenges when you're not working with mice or humans or normal experimental models. But B, because of the work that we were doing previously. And the work I was doing, I was working on an organism. It's got quite a common name. It's called chlamydia. And when you work on chlamydia, some people give you a certain look. And when you say you're working on chlamydia in sheep, you can get that kind of reaction quite often. But it's a really important disease. And it's a really economically important disease in the UK. And it's the biggest cause of infectious fetal loss in sheep in the UK. It's known as OEA. And Peter asked specifically if I could put as many grotty pictures in this as possible. And so this is actually a picture of a sheep placenta, and it's not a healthy sheep placenta. It's not supposed to be. It's infected, it's red, it's raw, and you see these little buttons there. They're called cotyledons. They're not like human placentas. That's where they fix onto the mother's uterus. And that is all inflammation. Okay. And what we showed was, and one of the reasons I wanted to highlight the previous work was the top panel up there. We did some cell lab work as well, where we showed that actually the cells of those cotyledons there, when they get infected, they produce those pro-inflammatory indicators. They produce those immune cells, the TNF alpha again. So there's a bit of a thing about that. They're not just bystanders. Everybody thought that they just were passive. They were very active and aggressive in trying to counter this infection. Unfortunately, that's what causes the disease. So it's very much an autoimmune disease caused by the infection. And that's pretty similar to nearly all the chlamydial infections in men, koalas, animals, whatever. So one of the big problems when you're working with this disease at the time was this. And it was really boring that people kept on saying, why the hell are you working on this? Because there's an effective vaccine available. And we come back to them and say, it might be effective, but A, there's never enough of it. And B, there's always cases of breakdown. And, and so it's not perfect by any stretch of the imagination. Why was this still remain such an important disease? And I'd been at a conference, and back then, you can tell how old it is. We'd sort of, I was in a conference in Spain, hard life, being a scientist, and somebody had said, well, we've sequenced the vaccine. We know what the vaccine genetic code is. And now we've got tools. We can see if the vaccine, we can tell the difference between the vaccine and the normal strains. And I'd got a load of samples through. And I said to my boss, David, can I have a look? And his answer was, well, you can have a look, but I don't think you'll find anything. And this, when it comes back to being a Yorkshireman and being slightly stubborn and pig ignorant, I went and had a look. Because you do. And fair play to David. And this is about as heavy as science as it's going to get. Please forgive me. So in this, so we had a look, and it's an assay where you amplify DNA. Okay. And it comes up with this band at 319 there. Now, if it's normal, you can cut it into bits. So this one, S26.3 and AB7, they're normal. But if it's mutated, it can't be cut. And the mutation was what was in the vaccine. It's what caused the vaccine not to be a vaccine, to be attenuated. And 1, 2, 3, 4, and 5 are field strains. So they were samples that we were sent from farmers. And so we demonstrated that the vaccine was the cause of the miscarriage in those animals. And we published a paper on it. And probably, as you can tell, it was not widely and wholly received, particularly by the pharmaceuticals companies that made the vaccine, and nor by the government itself. But we worked hard to stop disinformation. And this was the first time that I'd ever done anything really about impact. And I didn't know anything really about press releases or anything. So when a paper like this goes out, it's accompanied by a press release. And when you and the people who receive that can take it and they can take the text, and this is how the funder, the BBSRC, responded. This is word for word what was sent out. Vaccination still best option for controlling ovine and zoonotic abortion. Okay, link between vaccine and disease. The pharmaceutical press covered it really quite straight down the line. OEA vaccine implicated in case of disease. The Scotsman. I've been incredibly careful. I never spoke to the press in this. This was all from the same thing. And actually the text of the main body was exactly the same in all these cases. Now, I am very grateful that I didn't have to deal with them. I spoke to farmers and I spoke to vets, and I preferred speaking to farmers and vets, frankly, than journalists. But somebody did, and they are sat here. Because you want to talk about research impact, Maggie was our communications manager, and she's now my wife. And we met through this work. So I think that smashes any ref impact case study. Thank you. So, and Harry's here as well, our son. Right. So that work did have significant impact. And actually it was legislated in Germany. It's mandatory to test for that now. And there was a big pharmacosurveillance system put in place, which is still at Morden. But as a jobbing postdoc, and I'll say that a jobbing postdoc, it's not meant to be disparaging. I was going from contract to contract. I wanted something to define me. And so I started to think, what can I do a bit different? And I started with thinking, I like cows. And cows are economically a bit more important than sheep. So if you lose a pregnancy, the cow's worth a lot more money. So maybe we can actually get somewhere with this. And I'd also come across a new type of infection called chlamydia-related organisms. They're like first, second, third cousins of the standard chlamydias. And so I went again to David and said, can I have a look? And again, he said, don't think you'll find anything, but go on. And I did, and I looked, and I'm not going to go into too much detail, but we started looking, and being at a place like Morden where you have lots of diagnostic samples that you can just pull out the freezer that people have looked at but not found anything, you can sort of do this work. And this is just an analysis, an assay, where you amplify a bit of DNA of the bug of interest, and if it glows like that, it's positive, and if it's black, it's negative. And in about 25% of the animals that we looked in, we found these bugs. And we did that with Scottish samples, English samples, Irish samples, even Spanish samples, because people were sending us samples from around the world to have a look at. And this was just a picture. I'm quite proud of this picture. It's quite pretty. It's a cow placenta. And these individual brown dots are individual organisms labeled with an antibody against the parachlamydia. So that's what it looks like in the situ. And we got some money to do some work. And the best thing about that was it was the first time that I'd really been involved in writing my own work. And I got to write in what I wanted to do, from the cellular level to the animal level to big surveys. And the first bit of this was some work, and that's just an electron micrograph that I did of the organism itself. Waddley, it's a funny name. It was found in the Washington Animal Disease Diagnostics Lab, hence Waddle. Ego, I think, getting the better of them. But the reason I highlight this paper is not because of the science, but this was my first collaboration with Edinburgh Napier, with Peter and Mick through an MRes called Chris Coyle, who did some really good analysis as part of his MRes. Okay, so that was my first sort of real interaction with Edinburgh Napier. And you see where it's got me now. Now, just to fulfill the grotty picture criteria, I also thought I better include this, which was a bit of the whole animal study. And you can see the yellow stuff on that's a cow placenta. And that's full of these little bugs. And we managed to isolate it and then be able to stain it up with this brown stain. So anybody who's microbiology of interest, we did manage to fulfill something called Koch's postulates that's really important for a pathogen. It was really, really important. But probably in terms of where my career has gone lately, this result, which was actually quite a minor result, is probably one of the most important. So I think you can't take for granted what you're doing. And the reason it's the most important, this is Coxiella burnettii. And it's an organism that is endemic in UK cows. And I'd asked if we could analyse the milk samples for this organism to see if we had what levels we had across the UK. And it's not particularly this result, although I did have to fight to do this. And I had to fight to do it because it's a zoonotic disease. And people were again panicking a little bit about what the result would be. So a zoonotic disease is a disease that passes from animals to people. Okay, and it can cause problems in long-term health problems in people, but also pregnancy complications. But it wasn't so much that, it was because not many people were working on it at the time. And as a result, I was contacted by a colleague, Joe Halliday from the University of Glasgow, to analyse samples from Tanzania, so livestock samples for Q fever. And straight away, my ears pricked up because I thought with the zoonotic context, you know, people in this country, how many people have, it's a rhetorical question, please, so don't all put your hands up. How many people have had intimate, let's not use that term, close contact with a sheep or a cow recently? Probably not many, okay? But in many parts of sub-Saharan Africa, they're living alongside their animals on a daily basis. So actually that zoonosis and that risk to people was really interesting to me, was actually transferring what I'd learned in the UK into an African context. And I'm just going to throw out some results which will flash quite quickly because I think they're quite dry, but it's worthwhile noticed. And we got some money, and it was the first grant that I held at Napier to do some work to look at the prevalence of these organisms. And we found that Coxiella actually was one of the main causes of the fetal loss in the livestock in Tanzania. Okay, and this was actually all those N=6 was all goats. So that was really important. But I really got I hate to use the term juices flowing, but I really wanted to expand this work. And I wanted to expand this work on my own. This was all through Glasgow. And so I was so fortunate that about a month, maybe even 3 weeks before COVID lockdown, I went to Nairobi and I've traveled to Kenya to visit somebody I'd met before, a collaborator from the University of Nairobi, Thumbi Mwangi, who had a study site in the north of the country. So we traveled all the way in the north, a place called Marsabit. And he had a USAID project looking at how you could feed animals, lactating animals, more food, and how that milk that they produce, the increase in milk, would affect the children's health in that area. And actually it got very good results, nutritionally wise. Those children tended to develop quicker, even within a couple of years. It was a great project. But they'd also taken blood samples. We also wanted to know what diseases were circulating in those areas. And so, again, fortunately, we managed to keep this work going during COVID. And what we found was actually, yes, you can see there's quite a lot of it around. So about 70% of the animals were positive, had shown some level of exposure to the disease. And in people, 11% of women and 13% of children. You notice there's no men on there, but these are pastoralists, and the men were mainly with the camels off traveling at the time of this study. So it's quite high. But I was also working, not exclusively in Africa, I was also working in Edinburgh on a study of miscarriage in women at the Royal Infirmary with the Centre for Reproductive Health. And we were looking mainly at chlamydia. I'm not showing those results today, but we also looked at Coxiella exposure. And just to sort of reaffirm some of my thoughts about why I was interested in looking at zoonotic transmission in Africa was that we found only 0.8% of the women in our study. Much smaller study, obviously. It's not a rural population. It's quite a self-selecting population at the Royal, but a much lower percentage of people are exposed to those zoonoses in this country because they're not exposed daily to these animals. But again, the question I had was, I had lots of questions, and some of the work that Thumby had done there showing that that food supply makes a really big difference to children's health and people's health, made me start thinking a little bit wider. And I took over during COVID the co-lead of the Animal Health and Greenhouse Gas Network, Intensity Network from the Global Research Alliance. And so I started to think a little bit more about animal productivity. And this is not my work, but it just backs up what I was thinking there about what are we doing in terms of how we're producing food. And this is just a picture very quickly. It just shows that the population in Africa is doubling between 2010 and 2050. However, the need for animal source foods is tripling over that period of time. Now, one of the ways you can sort of do that, you can sort of countermeasure that, is you can just keep on increasing the number of animals, but it's not particularly good to the planet. It's not particularly good. It's not grazing land. They produce lots of emissions. They can affect biodiversity. There's lots of problems with that. And theoretically, we've been talking for a long time about what the problems of sick cows are. Sick cows, not just sick cows, sick animals in production, in livestock and food. And generally speaking, animals react the same way to illness that we do. You know, you tend not to eat as much, you tend to be a bit lethargic. Your immune system's fighting an infection, okay? And you produce less food. And so as a result, they produce less food. And so they produce more emissions, greenhouse gas emissions, and they produce less food. And so we started working. I managed to get some money with the director of this centre, the Mazingira Centre in Nairobi. And if we go back to that work I was doing in Tanzania, part of the money was, let's try and quantify what the effects of disease actually are in sub-Saharan Africa. So we went back to that data that we'd already collected and we modelled it, and we found actually from that reproductive loss data from Tanzania that we're losing enough food to feed 650,000 people on an annual basis. And we're raising our emissions. And we actually got in touch with Thumby and we got some data from him on calf mortality. And we found in Kenya, we're losing food just from calf mortality that would be enough to feed 3.3 million people per year. And that's huge. And we've just been in policy discussions, actually. Last week, I just came back from Nairobi, where we started presenting these figures to government officials to try and include them in some of their climate commitments. And grateful for that, with lots of people externally that are far cleverer than me to do the maths on that. Maths has never been my strong point. But I'm just going to run through quickly. We're also doing some field work in Kenya. So we're also training farmers actually in best practice and hygiene, because we're finding that there's a lot of infection of these animals' udders, which means that bacteria are coming through in the milk and there's a lot of inflammation in those. And that's ongoing work. And we're hoping to expand that to 6 more countries now, including South America and Southeast Asia. So we're really excited about what we're doing there. But I'm going to finish. I've got a couple more slides, but I'm going to finish because I think I've presented my research journey as a straight line. It has not been a straight line. It has been quite a rocky road with plenty of dead ends. and plenty of failure. And I think all through my life, I think that starting in sport did actually quite a good job of grounding me in the idea of failure. But also that failure often can breed creativity. And I think when you work with microbes, and microbes and infections cross borders. They're not really bothered sometimes about what species that you're in. And so sometimes during that gap and sometimes during those cul-de-sacs, I've had some opportunities to work on some very weird and wonderful organisms. And I'm quite proud, particularly the Komodo dragons. Looking at infections there. And I've taken that through to Napier. And believe it or not, so from when I first arrived, I had the money on the to do the work with the livestock, but working on sexual health in men and women with Sally Brown when she was here. Using these little beasties to test new antimicrobials and pathogenesis. Believe it or not, endosymbionts of hoverflies with Gavin, I don't know if Gavin's here, with Gavin Ballantyne with the ecology. Many people won't realise, recently, looking at the causes of nasal and ocular disease in Siamese crocodiles, captive Siamese crocodiles. And finally, I'm incredibly proud of newest PhD student, Georgina Cole's project, working on flapper skate and looking at health parameters and infections within these quite endangered species. So I think sometimes when we fail, actually it can breed a little bit of creativity. And also, let's just be flexible at times. Oh, sorry. Edinburgh Napier's disappeared off here. I don't know why Edinburgh Napier's disappeared off here. It was on the file that I uploaded. So nobody at Edinburgh Napier. So what I wanted to finish with briefly is that one of the reasons for showing all those papers is I'm never alone in all those papers. There's always, at least usually nowadays, half a dozen other people on there, and often from about half a dozen different countries or institutions. And I'm so grateful for all my colleagues who are not here. You see, if I've forgotten anybody now, this is a great excuse. I can say you were on there on the original file. Students who often do the hard yards on there, and my colleagues at Edinburgh and Glasgow, and also in Nairobi and on the methane work. Just before I finish, I'm not going to do the usual with the funders and just say I'm grateful. I have got to say that the journey of my research journey has actually been a journey of funders. We always used to rely on BBSRC, UKRI, but now a lot of the work we're starting to drift in this cloud towards these philanthropic funders, and starting to be a bit more creative with where we're looking for the funding for this has been really key to a lot of the work lately. And finally, just a plea, I suppose, to a lot of people. Networks have been huge in my career. And some of these have been funded stuff, but a couple of these are Edinburgh based. And I think they've been vital because this is all, as Eva said earlier, you need to make friends before you influence people, I suppose. So just finish it up. I've got to thank everybody, family, friends for all their support. I have not put pictures up. Maggie and Harry have been embarrassed enough already. I'm sure Kieran, who is sat there, would not want his photo on this screen. So thank you.